Merhabalar herkese. Hepiniz IEFTalks Webinarları'na hoş geldiniz. Bugün University of Exeter'de biyobilim programları hakkında öğrenmeniz gereken tüm detayları Sam ve Robert'tan dinliyor olacağız. Lütfen sorularınızı soru cevap bölümünden yönlendirmeyi unutmayın. Yes, Sam, the stage is yours now.
Thank you very much, Zainab. Welcome all of you. Just to be aware, the only thing I can see is myself and my screen. So I'm going to picture you all really excited, glued to your screen, looking at every single image on my slides and enjoying it. So if that's not the case, if you could say in the chat, and I'm sure Zainab will let me know that you're suitably uninterested or you want me to talk about something else. I believe that we are planning to have questions at the end. Let me just show you a rough summary of this afternoon. So approximately 45 minutes. I say 40, but the thing about academics is whilst we're really good at our subjects, the thing that we're not good at is timekeeping. So if I say 40 minutes, perhaps therefore it will run to a 45-minute time allocation. The last 15 minutes or so are set for questions. I believe that you know to put your questions in the chat. We'll happily get through as many as possible. If you're absolutely desperate and you cannot wait till the end of the lecture, perhaps something can be arranged by the chair. We know sometimes that happens. And also, this is probably a good opportunity to introduce my colleague, Dr. Rob Ellis, who you may see on the screen as well at the moment. He will be giving an example lecture. So he will have to leave maybe a few minutes early because he has other responsibilities. However, you've got me for the full time. So before we get into it, I wanted to briefly explain why this is a two-person job. The reason being is that Rob, as you will see, is a biologist. He spends a lot of time in wetsuits in the sea. He wears a normal suit in the office. But he's a biologist. He's interested in creatures and organisms and things like that. And I'm a chemist. So before coming to Exeter, I was really interested in ceramics, which is about as boring as it sounds. However, moving to Exeter, working in what's known as a bioscience department, it's really increased my landscape of things that I can look at. So the kind of places that I've worked at Oxford University. I'm sure you've heard of that one. I was a researcher at Princeton University, another one that you've probably heard of. I've also worked at Manchester here in England, Stockholm in Sweden, a place called KAIST in South Korea, which is like the Asian MIT. So you have robots walking around the place, that kind of thing. Until quite recently as well, I had a guest professorship at the University of Shanghai Tech. So I've been around a bit. And I can honestly say of all the places that I've worked, Exeter stands out in two main ways. The first one is that it gives me access to all of these extra samples and all of these new ideas. Because if you're in a chemistry department or a biochemistry department, you're somewhat limited at the scope of what you can study. And this is as much true for academics as it is for students. And that's something that we'll be coming to at the end of this talk. And then the second thing is the level of student support. All the places I've worked, Exeter's by far the best. Okay, so if you can't stay for the rest of the talk, that's probably the most important things about why Exeter is so great. However, I hope you will stay. Maybe many of you are not quite sure about what you want to study. The kind of students that we accept are the top tier students, those who've got very, very good grades. For our A-level system, that's things like an AAB. Where we expect things like chemistry and maths and so on, depending on the subject. I'm guessing as most of you are on your laptops or something like that, I recommend that you go give us an internet search while we speak, and then you can ask us about grades and things like that towards the end. So that's your homework to be doing while I'm speaking, because I know that you can focus on the two things at once. So most of you are probably going to get really good grades and you're thinking, well, why go and study? Why go and study bioscience? You probably had the question or the conversation with your parents where they've probably said something to you like, why don't you be a dentist? Why don't you be a doctor? Why don't you be an engineer or perhaps a veterinarian? All very good career choices. Your parents probably like the idea because it brings in good money and it's stable. And if the fact that you're on this talk tells me that you haven't yet made up your mind, and it's okay not to know what to do. I'm sure Rob will echo when I say that we're curious about how the world works and how we can help, and kind of not knowing where we're going or necessarily what we want to do can be a superpower, provided you have that frame of mind of wanting to learn about how things work. And I've got some good news for you. If you're that way inclined, you're never going to run out of things to understand, partly because the world is so complicated and there is so much to discover, but partly because the things that you can learn about, the practical things that you can learn about and help people, they're always changing. Back in the 1960s, there was a book released called The Population Bomb, which said that the population of the world won't survive over 2 billion people. And that calculation was made by counting up all the nitrogen that was in the soils and saying, this is how much nitrogen there is in the soil, and this is how much food we need, therefore we can't go above 2 billion. Now, that was clearly false. That was clearly hubris on the part of the people predicting it because they forgot one important factor, and that is the innovating nature of human beings. We have been able to increase that population to nearly 8 billion. So when people tell you that the world's dying and so forth, it's always been dying, and we've always been pushing back the boundaries of what's necessary. As we've grown our crops and our food sources, so too have we come across new challenges. I want to draw your attention to the pictures on the right here. This one especially, the sort of green leaf that you can see. That's known as a rice blast or banana blast. They affect both species. And they have the impressive title of being the only pathogen that's actually visible from space. So people in the International Space Station can actually see this pathogen infect crops. It's quite extraordinary, really. The reason being is that we have so much crops in the first place, but the reason that these pathogens are able to take hold is because of things like the Green Revolution, where we selected crops that were more appropriate to certain conditions, where we might have, and you may have studied this in geography, perhaps, where we have, for example, a field of 50 different species of rice. We replace those 50 species with the one that's most suited to that particular rice field. That sounds great, but another form of hubris. What happens when that crop gets infected? Well, if there were 50 species, perhaps some would survive and some wouldn't because that pathogen was tailored to one specific type of crop. But when you have the one crop over many, many, many acres, you can get these problems. So we have to come up with new solutions. And that's the kind of thing that you do with a degree in biological sciences or biochemistry. You help people, but rather than a doctor, which was probably one-to-one, you help people on economic scale through things like innovation, solving of problems. And that's something that we're very proud to help deliver on. There are plenty of challenges. It isn't just rice. It isn't just bananas. I asked a colleague of mine to come up with all the different things that we should be focusing on, and this is what we came up with. Many of you will think that's a frightening number of things to think about, but to be honest, honestly, we just think about that as an opportunity. So that's more of a chemistry lean perhaps. So I want to give a little bit of balance, and I want to give you an idea of the kind of things that we have as lectures. It isn't just all about pathogens and chemicals and things like that. So in a moment, I'm going to hand you over to Dr. Rob Ellis. Are you going to introduce yourself, Rob?
Yep, no, very happy to do that.
Okay,
Completely.
well, based on the fact that I can half talk and so can Rob, I'm going to pass over to you right now. Before I do so, Rob, if you just say next, I'll move on to the next slide as and when.
No problem. Thank you, Sam. Well, next. Yep. Hi, everyone. Well, my name's Dr. Robert Ellis. Yeah, sorry. Can you animate this slide so it all appears, Sam? So the bottom will, that's it. Perfect. So I'm a lecturer or a senior lecturer at the University of Exeter. And like Sam, I'm a really kind of inquisitive person and like to work out how things work, particularly aquatic organisms. So as Sam said, here's a picture of me in my typical attire. I don't often get suited up for work. I often wear wetsuits and I'm in the marine environment. And I really focus on how humans have adapted and affected the marine ecosystem and how that therefore translates and affects organisms in a range of different settings, one of which is aquaculture. So how we farm aquatic animals to provide enough food for us to eat. And Sam introduced that a little while ago. And it's one of the major challenges we've got in terms of the growing global population. How can we provide enough food? And seafood is one of the major sectors that's being affected. It's the fastest growing food sector, aquaculture, but it's one that's really impacted by climate change. So I teach at Exeter on a range of different things in marine biology and kind of animal physiology and understanding that nexus. And I did my PhD at the University of Plymouth. Unlike Sam, my career has been very southwest of the UK focused. Plymouth and the University of Exeter are where I've done all of my research and studying. I have traveled the world. I've done research in Antarctica, all 7 continents, working in a whole range of different environments. But I've always been based in the southwest of the UK, and I can really kind of support what Sam said about how incredible the place is. Next slide, please. So what I'm now going to do is give you a bit of a brief taster lecture on some of the research that I've kind of undertaken over the last decade or so, and some of the really important questions we're addressing. And I'm going to pose to you first a question, or at least a thought, and it's about the fact that I think scientists don't think about sex enough. If you could animate that, Sam. And I'm going to talk about why I think that's the case. And back in 2017, I did a research study looking at climate change and ocean acidification. And that showed that less than 4% of studies looking at climate change actually think about whether males and females are affected differently by climate change, which means we could be missing a large part of the picture. Now, I'm going to go right to the end of that story with sorry, if you go back one slide, Sam, with the end of the story with those 2 images on the right of your slide there, talking about the outputs and the impact that this research has had. I was part of a review panel for the European Commission. So we really drove EU policy on how science is undertaken as part of this research. And we actually published a paper in Nature, which is considered the pinnacle of scientific research. So this really shows kind of the provenance of the research we've undertaken in our lab, all about asking that one question: do scientists think enough about sex? Next slide, please. So what we're going to learn over the next sort of 10, 15 minutes max is thinking about what is sex? Why are we considering sex? Why do we need to think sex is important? I want to highlight to you just how diverse sex is in the non-human biological sense. So thinking outside of our typical scenarios, how strange, how interesting sex can be. And then lastly, I want to show how actually working at Exeter and studying at Exeter, you can help answer some of the questions that feed into things like those EU policies and that Nature paper. Next slide, please. So what I want you to do is have a look at this graph. And essentially, what you can see here is a real data set. So this is copepods. So you can see the animals on the left of your screen are very small marine crustacea. There's billions of them in the ocean. They're some of the most diverse organisms. They're really critically important for the ocean's function. They're a food source for whales, for example. There's an experiment undertaken looking at climate change, so how much CO2 is in our ocean and how it affects these small organisms. Now, on the bottom of the graph, you can see CO2, high and low. So that's today is low CO2. A future ocean in 100 years is high CO2. On the y-axis, you can see something called respiration rate. That's how much oxygen those animals take out of the ocean to undertake their normal function, okay? And if you look at the graph, you've got some green dots and a flat line. Essentially, what that graph would tell you, if you were to interpret it, is there is no effect of CO2 on that respiration rate in these organisms. You would interpret scientifically, there's no effect of climate change. If you could jump through to the next slide, Sam.
Before I do, Rob, how big are those organisms?
They're about this. They can get from about the size of your small fingernail, kind of a little bit bigger than that. They can be much smaller than that. So they're tiny, tiny creatures. Sorry, I should have said, yeah. They're really, they're in the plankton. They're zooplankton, they're called, and they're very, very small.
Okay.
So What I'm doing now is the same graph, okay? But what we've done in this example, as you can see, the dots have changed from green to orange and blue. Now, really interestingly, the green dots are individuals where we don't think about males and females. On the blue and orange dots, as we look here, the orange dots are males and the blue dots, or blue diamonds, I should say, are females. And you can see two really interesting things. Firstly, males and females are very different in terms of how much oxygen they use. If you look at the current conditions, low CO2, females use much less oxygen than males. But if you look at climate change, actually, females increase the amount of oxygen they're going to use in a future ocean, males decrease it. So really interestingly here, you've got a scenario where the response to climate change is different in the males and females, and they can respond in a completely opposite way. If we hadn't actually thought about sex, if we hadn't studied males and females separately, we would have no understanding of this. And yet it could be really important to help us model and understand future climate change conditions. Next slide, Sam. So now this is one of my favorite things, and it's thinking about what sex is, and I get to introduce one of my favorite organisms, and this is called a sea sapphire. Hopefully on your screen you can see a video of something that's a bit iridescent blue. You just get flashes of blue light every now and then. This is a sea sapphire. It's another type of copepod. It's incredible. Now, this is a male sea sapphire, really importantly. If you can see on the right-hand side of the slide, you've got two black boxes with two very different looking animals. The top one says male, and this is what you're looking at in the video. It's a blue sea sapphire, and it's got something called structural coloration. So the light shines through its carapace, which is its hard exoskeleton. And if it shines just right, the hexagonal cells within its carapace reflect that light back exactly where it came from as an iridescent blue color. However, if the organism moves, that light is dissipated and attenuated within inside the carapace, and therefore it looks almost invisible. It's an incredible example of how this organism is able to hide and camouflage unless it wants to be seen. Now, the picture below, the female, looks very different, and that's because it is. It has a completely different body plan. It lives in a different place. Males live on coral reefs on the bottom. Females live on fish. They actually attach to fish as a parasite. The reason the males flash like this is because it attracts females down when they need to mate. So if you look at the picture of the female, the blue sacs on the back are actually eggs. The female, unlike the males, has huge eyes. So they've evolved to have massive eyes to be able to see the males and come down and be attracted to the seafloor. Now, sex is actually any biological attribute that distinguishes between males and females. So it can be things like the presence of different gonads, the different gametes, so either eggs or sperm. It could be the reproductive anatomy. It could be your hormones. There's many different biological traits that differ between males and females on a whole range of different scales within the organism. Next slide, please, Sam. So, does sex really matter? We've shown in a climate change context, we don't really consider it very much, but actually there's some really clear examples of where understanding the difference between males and females is critical. Now, in humans, we're actually really kind of trained to think genetic sex is normal. So when our eggs and sperm mix, you're either turned into a boy or a girl because of XX or XY chromosomes. They're the DNA that determine whether you're a male or female. We think that sex is fixed at the birth, so you're either male or female your whole life, and anything outside of this is strange. Next slide, please. But in nature, it's way cooler than that and far more interesting. So in nature, over 30% of animals display hermaphroditism. I'll explain what that is in a minute, but essentially it means an animal, one organism, can be both functionally, biologically male and female. Sex change is prevalent, so an animal can be male at one time and then change to become a female, and we'll give some examples of that. And also what determines if you're male or female can be a whole range of different factors that are genetic, like humans, but could be due to the environment. And we're going to look at some really cool examples there too. Next slide, please. So hermaphrodites, as I said, organisms that can be both male and female at the same time. So the top in your picture here is something called a sea slug. They're some of the most amazing, flamboyant, exciting marine creatures. They're, again, very small, but they're amazing. They have some really cool features. There's one called the solar-powered sea slug. It eats algae, and it puts the chloroplasts into those appendages on its back, and then it can photosynthesize. It uses those plant cells in its back to photosynthesize and get its energy. How cool is that? But also, all sea slugs are hermaphrodites. They are both male and female. They can function as a male and a female at the same time. This means that an individual can deliver sperm to another organism and fertilize that organism's eggs at the same time as receiving sperm from an organism, and it has its own eggs that can be fertilized. So it can be both male and female. Now, some organisms are sequentially hermaphroditic. That means they're a male and a female at the same time, like our sea slug. Also, just behind Sam's head on your screen, there's a picture of a scallop. And you can see here, scallops are a really tasty seafood, and we know that you've got the orange and white bit at the bottom of the mussel, and that's actually called the roe. That's where the sperm and the eggs are. And the reason it's two-tone, white and orange, is because the orange is the eggs and the white is the sperm. So that scallop is also male and female simultaneously. Oysters, on the other hand, they change sex. So they typically start as male, and then at some point during their lifetime, they can change to become female, and they'll switch their entire biology to produce the different gamete. Next slide, please, Sam. Now, as I said, what determines sex? So if humans have genetic sex determination, your genes that you inherit from your parents determine if you're male or female, and that's just the same in carp. So carp are determined male and female by genetics. Next. Some organisms is entirely environmental. This is an Atlantic silverside, and the temperature at which that animal has been fertilized determines whether it's male or female completely. So it's all about the environment. Next. Or you can have a mix of the two. So this is sea bass, again, another really famous food fish that's really heavily produced in the Mediterranean. And it's really a tasty seafood. But again, a sea bass has some element of genetics, but the temperature, again, interacts to determine whether an individual becomes male or female. So it becomes far more complex than we ever give it a credit for, thinking about humans. Now, the environmental factors that can determine if an individual's male or female are really interesting. So flounder, if it's too hot or too cold, they become male. If it's just right, like Goldilocks, then they become female and they produce more eggs. In eels, it's density. If there's far too many animals, they produce more males. If there's not that many, they'll produce more females. In zebrafish, that's the stripy fish at the top, the one that has oxygen underneath it, they need good levels of oxygen to produce females, but if there's low oxygen, they produce males. Now, this is all because when the conditions are seen suboptimal and not ideal, they produce males because males can change the amount of genetic diversity in the population. But if conditions are perfect and there's lots of energy for growing, they invest in eggs because eggs are very expensive to produce. But it means if there's lots of energy, the juveniles that you produce can actually grow up and they can survive. So it's all about whether you need to adapt or if you want to invest in good conditions and have lots of ready growth. The example on the bottom you can see here is a clownfish, Nemo, and they've got a really interesting example through social status. So Sam, can you click on the next slide and see if we can get the video? The video will play automatically. So clownfish, they have a social structure where they have one dominant female at the top of the social structure, and then underneath it, it's all subordinate males. Now, as you saw in that video there, they're very territorial. That female just bit the tail of a shark to scare it away from its family. You can see here, thanks Sam for showing it again. She's swimming away from the coral reef and bites the shark's tail to scare it away and to protect her family. Now, that's a very bold move. She's a very small fish compared to that shark who could quite happily eat her. Now, if she was eaten, what would happen is she would be removed from the population, and the next male in that family structure changes sex to become a female, and all the other individuals underneath move up one rung on the hierarchy in that family. So they are all male. This means one thing, however, and I'm sure you've all seen Finding Nemo, or at least I hope you've heard of it. This is Disney's film where where Nemo, the small juvenile clownfish, his mum's eaten by a barracuda at the very start of the film, and he goes off on a huge journey, and the dad actually chases him to see where he's gone. In reality, what would have happened is as the mum's removed, Nemo's dad would have become his mum, and Nemo would have become the dominant male in the family and then started mating with his dad. Biologically, far more interesting, slightly less suitable for a Disney audience, I think you might agree. Next slide, please.
I'd say definitely not suitable for a family audience, but definitely suitable for an adult wanting to study about how the world works at university. That's for sure.
Indeed. Right, next slide. One last example I'm going to really quickly give you is that of turtles. And very similarly to fish, turtles' sex is determined by the temperature at which their eggs incubate within the sand. As you can see here, if it's too hot, you have more females. If it's colder, you have more males. This poses a really big problem with climate change because as the climate's warming, the nests are warming up and you're having less and less males and more females. And actually some populations are becoming locally extinct because the temperature is too warm to support balanced population ratios. So this kind of impact of environment on sex can have a really significant impact for population survival. Next slide, please, Sam. So now kind of thinking about all of those examples, I want to come back to this example here and think, okay, now we've covered really that diversity of different impacts of sex. I want to ask that same question again. Do you think sex is important? Hopefully, I've given you some really cool examples of just how interesting it can be, but also how important it is to think about sex in terms of climate change and how organisms adapt to their environment. Next slide, please. As I said, it is, I believe really passionately, it's massively important. But the truth is, we still don't know just how. Only 4% of studies, as I introduced at the beginning, study sex in a climate change context. So we really need to do more research. And that's where you can come in. Next slide, please. If you come and study at biosciences, I've got projects undertaken in the lab that I'm working on with students that are asking these kinds of questions. We're running research that's research-led, actually I'm kind of integrating with my students, looking at those questions. How do copepods get affected? How do mussels get affected? So if you came to Exeter, you could be doing research during your degree program to help us answer those questions that are at the forefront of the field. And that's just my area of interest. All of the academics are doing similar things, and you can embed yourself in a whole range of topics to answer those questions and be doing really key cutting edge research. I believe that's the end of my presentation, Sam. So I'm going to pass it back over to you. I want to say thanks to everyone for listening. And again, we can answer questions, but I'm going to pass back to Sam now for a bit more of a generalized overview of our teaching portfolio. So thank you.
Thanks, Rob. I learned something too. Depending on how much time we've got left, I'd love to show students some of my research, which I'm not sure how much you know, Rob, but my hypothesis is that things like butterfly wings and other iridescent materials are actually crystals, called crystals. So I have some really nice electron microscope images or micrographs of that kind of structure. However, we do have to cover the basics. That's why we're here. I'm sure before you decide what you as an audience are going to do in terms of further education, if you choose further education, you will attend many of these kinds of talks where you see slides on grades and so on. Hopefully, though, we've given you something else as well. Hopefully, something to consider about the sex of species and how that affects development in various novel scenarios, such as the ones that we find ourselves in now. Okay, here it is, the obligatory stats and so forth. So a little bit about Exeter. We are 20 in the UK for biological science. We have very high levels of student satisfaction, very high indeed. And we, as Rob said, we do research-inspired teaching. So almost everybody who will lecture you will be involved in research in some way. There's very, very few academics who get out of that. It's one of the things that we pride ourselves on. Okay, so more about that in a moment. We fall under what's known as HLS, Health and Life Science, of which there are these other disciplines. Sometimes we get mistaken for medical science. Sometimes we get mistaken for conservation, but we are our very own department where we study biology and chemistry and how they interact in lots and lots of varied ways. I would say of all the departments, we probably have the most varied selection of modules available to students. So if you're not quite sure what it is that you're interested in, you could do a lot worse than choosing a biosciences degree at Exeter because there is so many options to experiment with. Here's some of the examples of our research that we get involved in. Okay, so here's some examples. We often do make the news. We have great relationships with people like the BBC. Very common to hear my colleagues on in the morning. I remember texting a friend saying, "Oh, well done, I heard you on the Today program this morning on the radio." And he said, "Yeah, I was tying my kid's shoelaces while I was on the radio." And I thought, wow, that's pretty amazing. You know, we are very good at speaking to the public about scientific issues. Okay, I'm just frightfully aware of the time, so I'm just going to skip a bit because I want to focus on the kinds of things that you're going to be interested in specifically. So here's a picture of our campus. We have 4 campuses. We have one in Cornwall as well, but we're both based at what we call Streatham. I'm speaking to you from this building here. Just around here. These are all research institutes. That's a theatre. This is called the Arcs. This is where we do a lot of research. All the people that you see here, apart from this person, they're all students. So they're all people who are doing research in their final year as part of their degree. It's quite an impressive place, and it's only one of 6 of our facilities that we have. So students are placed all over. An incredible place. Beneath our feet, it exists, and there's about 15,000 fish tanks, which is quite extraordinary, I think. We also have, if you'll excuse me, a fantastic city. We have a great cathedral. We're very, very close to the sea, very, very close to the moor. 50% of all Bronze Age artifacts in Europe are still on Dartmoor, which is very accessible from where we are. It's quite extraordinary. If you've seen things like Stonehenge, there are stone circles all over the place, which you can just go and find. But it is a great city. Specifically related to teaching, I wanted to show you this slide because we're what's called gold for our teaching, which is the highest one can be. And it's something that we're really proud of. All English universities get graded, and we're graded at the highest, so we're very happy with that. So that's perhaps one to write down or make note of. I mentioned about the 3rd year project. Here's some more examples. It's not just all fish tanks. Here's a leaf pathogen that someone's imaged with something called a scanning disc laser confocal microscope. We have people doing cytometry, all kinds of wonderful things. And that's part of your final year. And we basically gear you up from your 1st year to take part in this project. Here's some more examples of the kind of things that students look at. And here's our other facilities. So generally our degrees are 3 years. If you're studying either biological sciences or biochemistry, those are our 2 programs. If you're an undergraduate. If you're interested in master's courses, do let me know. I'll happily talk about those. But yeah, generally they're 3 years. However, we do have 4-year variants as well, which might be something to think about, and I'll talk about those. And I think once we've got that far, we'll probably call it a day and open the floor to questions. So if you're doing biosciences, biological sciences, you'll be having a personal tutor. So between 4 and 6 students are partnered with a tutor, and you'll have that through your academic career, and they'll help you with things like essay writing and applying for jobs and stuff like that. So that's fantastic. I know many of our contemporaries, it's around 40 to 1 students to tutors. So that's something that we're very proud of. And we also offer 4-year conversions with professional placement. So instead of doing a 3-year degree, you do 2 years, and then your 3rd year you go out and study at some places like this. So GSK, for example, CFAS, and you can do that both with biochemistry and biological sciences. Or you can do study abroad, which is something I run, and it's something that we're very proud of. We have destinations all over the place, constantly changing. But my students are just getting ready to go out now. One of them's going to Montana State in America. Another one's going to Aarhus in Denmark. And the nice thing about this is that you only pay, I say only, 50% of the standard tuition fee. So if you're If you're looking to come to the UK, but you also want an experience of a university in another country, then that's probably something to think about. Here's an example of some of the courses that are available for biological sciences. As you can see, they're extremely varied. I don't expect you to remember them all. They are all on our website. And I would say, as a piece of advice to all students who are thinking about going to a university, another piece of homework is to go and look at the modules that we teach and look past the names. So you may see, for example, energy metabolism. Many places might have that subject, but go and look at who teaches on it, because it will all be very, very different. We make our own exam papers, we make our own assessments, so we teach the things that we think are important, and many of your teachers will be like Dr. Rob Ellis and have certain passions and interests, so make sure those overlap with what it is that you are interested in. We also run field courses as well. Now, I can't really talk about this because as a chemist, these field courses are mostly for biological science students. I keep asking if I can take my test tubes out there, but that's not for biochemists, I'm afraid. If you are interested in biochemistry, it's a very similar setup. We have the 3- and 4-year variants, but as you can see, our years are very different. This is what your first year looks like. So some of these you probably recognize, some of them you won't. And that's because we recognize that not just Turkish students, but all of our students from all over the world have probably had different exam boards. Some have done A-levels, some have done IBs, things like that. So we're very happy to accommodate that by making sure that we cover all the basics. So everyone's at the same, and I'm going to use a bit of educational theory here, but everyone's at the same luminary level. So just to make sure that if you have perhaps a deficiency, a comparative deficiency in your studies up to the age of 18, we can make sure everyone reaches the same level so you won't get left behind. We have many options in the second year, as you can see. One of them that I haven't included, which you might be interested in, is academic English, which is run by the Into department. And they are a special department here at Exeter that deal specifically for international students whose English is not their first language, and they run an excellent English for Academics course. We have fantastic feedback from our students that take this, and many of our students do. And when you're coming to Exeter, you'll be alongside other international students, and you'll have your tutor, and if you're struggling with your English and things like that, there are many. If you speak to your tutor, there are many things that we can help you with, and one of those is that particular course. And then here you go, your third year, and you see there on the left, research project. Note the box there is much bigger than the others. That's not by chance. That's because it forms such a big part of your final degree grade. Now, I'm sure you're not thinking about that right now, but when students arrive, that is something that's really important to them. We also have flexibility, so many people study languages. As long as it doesn't interfere with the timetable, we're very happy to support you. And more people are doing modules at the business school. A lot of international students are very interested in that. Perhaps they want to start a business when they get back to their home country. So that's something that they do. And if you get enough courses under your belt, enough courses, a number of credits, you can often get an extra thing attached on your degree title. So a proficiency in, for example, Russian or business or what have you. Okay, I believe I'm up to my time, so I'm going to stop my presentation now. Hopefully you can still see my screen. Maybe I should stop that as well. Let's see.
At the minute, Sam, we can just see yourself, not the screen.
Oh, that's great. Okay. Nice to see you, Rob. I'm glad you're still here.
Yeah. Okay, it was not. Oh, there we go. I was going to say it's not letting me turn my video back on, but there we go.
That's great. Could the chair make sure we can see both mine and Rob's faces? Because I think we're ready for questions now, if you want to override my control.
Yes, thank you very much for the great presentation, Robert and Sam. So we have a couple of questions in the Q&A section. Do you want me to read them out loud to you?
Yes, that would be nice. Thank you.
Okay, And our first question is from Tuna, and he's asking, what technologies in biosciences have the most potential to shape the future, and how is Exeter preparing students to be part of that change?
Wow, that's such a great question. Well, that's something that we ask ourselves all the time. So one of the things about being a researcher is that you're constantly having to justify your job to the people who give you your money. So every probably about somewhere between 25 and 33% of your time is spent writing for grants. So the people that you write the grants to have a very keen eye about where the future should go. And you're basically put in a big pool of other applicants and you have to decide, they have to decide what's important. So to answer your question, the way that we go about it is through brutal competitive arguments between different researchers trying to get money. And of course, the most innovative, the most clever, the most forward-thinking academics are the ones that actually get the grants and succeed. So if you take our department, for example, we have lots of traditional forms of technology. I say traditional, but they're still pretty cutting edge. So we have, for example, a sequencing service that can sequence genomes extremely quick. That was such an incredible thing to consider 20 years ago. But we also have much more novel ideas. So something that we're looking at is what we call lab on a chip, or how we consider running experiments. Because if you're working in biology, there are so many factors. You need to repeat your experiments a lot of times to get statistical answers. So we have machines that basically use AI and a microscope to look at cells, look at small organisms, and it basically sorts those organisms by things like size, shape, color, reflectiveness, how much they're wiggling around, and basically runs them off into different channels where we can then, for example, introduce stimuli into the environment and then sort them again. So this kind of like high throughput sorting of small organisms using AI to image them, that's something that's really important. Another thing that we haven't touched on at all is something called tomography. Now, many of you have probably had an X-ray, and you know what an X-ray looks like, even if you haven't had one. We would technically refer to that as a projection, because it's a 2D image where you're not looking at your hand, you're looking at a mass contrast of your hand. Well, we have microscopes that, instead of using X-rays, use electrons, and they can take these projections down to the picometer. And if you're trying to imagine what a picometer is, it's like the width. It's smaller than the width of an atom, right? It's really, really small. No, sorry, not the width of an atom, the distance between atoms, right? It's very, very small. And you can take these projections, move your sample around so you get different projections. And then the tomography part is when you then condense all of those images together in a computer and out spits a 3D model. Now, when I was your age, we were very excited about this because we were using it on things like things like skulls and fingernails and archaeological fragments. Whereas now, because we've got it down to this incredibly high resolution, we're able to move proteins and parts of proteins. And now we can even see these proteins interacting with other proteins. So it's my belief very, very soon from now, we'll be able to image a whole cell and see all the interactions play out. And I think that's pretty exciting.
Yes, thank you very much.
I was going to, sorry, Zainab, I was going to very quickly jump in. I've looked at the questions. I'm going to have to go in 5 minutes. There's two
Yes.
at the bottom that are specifically about the sex part of the lecture.
Oh,
So I was
yeah.
going to very quickly address those two. Sam's amazing, and he'll be able to answer all of the others. But
Okay.
but those two, thought I'd just quickly give you an insight into those two questions. Again, they're great questions. So the first one is from somebody anonymous, and it says, can we say male and females are always completely opposite? Absolutely not, of course. There's so many different things that are very similar between males and females, and there's lots of things that differ. The point from a biological context is we need to study this and consider it to know where those differences lie and where those differences are important. In some contexts, the difference between male and female are completely irrelevant and absolutely of no importance whatsoever. In some places, the difference between male and females is quite important, and actually understanding that. I'm going to give you a very quick example of where that might be important. Well, two. One is with crash test dummies in the US. Crash test dummies were always on average man, and that meant from a period of 2008 to 2018, women were 45 times more likely to have serious injuries in car crashes because seatbelts weren't designed for their bodies. Isn't that nuts? Conversely, there's drugs out there that really is targeted and only tested on males. There's one called desmopressin, which is all about how much water you retain. Now, really interestingly about desmopressin, it acts upon some transporters in cells that are coded for in the part of the X chromosome that would usually be silenced because females obviously have XX chromosomes, males have XY. And therefore, this part of the chromosome actually escapes that silencing. So females, as a result, are twice as receptive to the drug as males. So if you give females the same dose of that drug, they have twice as strong a response, which is actually quite dangerous. So there's something there again. We tested the drug, we developed it in males, and therefore it was more dangerous for females inadvertently. So no, males and females are definitely not always kind of opposite, but absolutely it's really important to consider where those differences are. The second question, it says, do you think with evolution, humans can change their sex as well, like oysters? They do it through surgery, but I mean as biologically. Now, that's a really big philosophical question. And there's an argument within biology that all organisms are moving away from typical sex-determining systems, and males are actually evolutionarily not advantageous. That's a whole big picture, and we're talking millennia for things like that to happen. But actually, a lot of what I teach through my biosciences research is we need to consider biosciences beyond the binary. A simple binary classification of males and females isn't appropriate. I don't think anywhere, like, again, that we're on a spectrum where you've got males and females at each end. All of the research is showing, if we look at nature, I think sex is actually a continuum. And I think we're only starting to understand the diversity out there because we haven't been looking quite as much. Now, there's examples for a good friend of mine works in a biomedical lab in Wales. And he said there's people coming in, a 70-year-old man who'd passed away. They had the autopsy afterwards to see what had caused death. Nothing to do with what had caused death, but there was this 70-year-old man had a perfectly formed pair of ovaries. Completely unawares to that person, his family, anybody biologically, the doctors did not know until they actually had a look. And actually, there's lots of things. Now, as I said earlier, sex is about your ability to produce eggs or sperm, or it's the reproductive anatomy, but there's lots of things. It could be the levels of hormones in your body. And there's lots of really high profile examples with athletes, female athletes that produce too much testosterone that are being inhibited from competing because they're deemed as what's called intersex. They have traits between male and female. I think that's actually probably much more prevalent in nature than we appreciate. So I think really whilst the evolutionary thing is going to be happening on a very slow timescale, far slower than we'll ever be able to appreciate ourselves, I think society needs to evolve in what we consider the importance of sex and the different levels at which sex can be diverse in society. And I think it could be really important in terms of our acceptance of things that perhaps don't seem quite as normal to us as we might like to think. And there's a lot of things going on politically. I'm not going to talk about different regions, but there's certainly political leaders that are very vocal on this in terms of their kind of their thoughts on that differentiation and how we classify individuals. But I think actually biology and nature shows us things are much more diverse than we actually appreciate. And it's often because we haven't got the systems to study them non-invasively to really get that full appreciation. So hopefully that's kind of answered those two questions.
I'm so glad you answered those, Rob. Everyone has an opinion on these things, but so few people have a qualified one. And I would definitely form into the latter rather than the former category there. So thank you for saving me.
No problem at all. With that, I'm really sorry. I'm going to have to step out, but I'm going to leave you in Sam's very capable hands and say thank you so much for your time, everybody. I hope you've got what you needed. If there's any further questions that we can't answer in time, we're really happy to answer anything over email or follow up later. So, but no,
Absolutely.
Thank you for your time. And yeah, I hope you enjoyed it and look forward to welcoming you at Exeter, hopefully, at least some of you, if we've inspired you to consider applying. So thanks very much.
Thanks, Rob. Sadly, I can't see the questions. I'm not
Yes.
sure about that. So
I will be continuing. So our next question is from Buket, and she's asking, could you tell us more about the lab facilities and fieldwork opportunities students can access during their degree?
sure. So we have a main teaching labs here at Exeter, which can accommodate up to about 280 students. However, the total number on the lab can vary from anywhere from 12 to 280. We have fume hoods in there. We have various FTIR, so Fourier transform infrared spectrometers. We have UV-vis spectrophotometers, plenty of skulls and things like that. I'm sure Rob could tell you about the dissection labs. However, handling more with chemistry, we have the standard chemistry kits. We also have a mass spec GCMS and proton NMR and a confocal microscope for students to use. My particular things that I teach first year chemistry. In my module, we get students in the lab every week, and we start with atoms, and then we build up to molecules, complexes, and then we run various thermodynamic and kinetic experiments. As well as that, when it comes to students in for their research, they have access to the same labs as our researchers. Obviously, you're paired with a particular one. So whether, for example, that is the ARC, the fish tanks, it could also be the chemistry labs as well. So we have a synthetic lab for organic chemists, or any of the other microbiology labs, things like that, the microscopes that we have. So it's really nice, actually. One of our buildings here, all the labs are on one side and all the offices are on the other, and it's all glass. And if you go in on a term time, you can look up and you can see all the final year students beavering away and working hard in their particular labs.
Yes, thank you very much. And next question is from Tuna. Is there any hands-on experience in the first year, or is practical work mostly concentrated in later years?
From term 1, week 1, we get you in the lab. Because we put such a high focus on your final research year, plus, well, your research project in your final year, we're trying to get you prepared from the start. So as well as getting you in the lab, it's probably worth pointing out as well that we get you writing. So I'm sure for most students, that's probably an audible groan if you had your microphones on. But the thing is, you need to be able to write. You need to be able to write dissertations. It's an important part of science. Being able to think requires you to write. So as well as lab work, we're also giving you all the other training for your final year. We would never expect students to just be able to do stuff. I'm very impressed, actually, compared to the places I've worked, how understanding and patient lecturers are with students, making sure that they are given the opportunity to test their skills and grow their skills before throwing them in a research environment.
Yes, thank you. And the next question is, do you encourage students to attend external conferences, seminars, or science activities during their studies?
So we have a couple of conferences on campus. We have a student conference at the end of each year. So we've got it in a couple of weeks now, where students give their talks about their research. We also have various society conferences as well that take place, and also undergraduates are invited to any of the research seminars that are given by the researchers or guest researchers. So we have a guest researcher every week, pretty much, from all over the place. If someone's coming to visit us about a grant or a piece of research, we make sure that they give us a talk. Surprisingly, undergraduates really don't get involved as much as we would like. But you'd be very welcome. If that question is also aimed at our postgraduates, then there are options open there. But I think this is undergraduates only. In terms of like funding and things like that for external conferences, no, unless it's part of your studies. And I realized as well, I glossed over a previous question about fieldwork. So there are a number of fieldwork modules which if you're doing biological sciences. Some of them have limited occupancy. So for example, every year students go to the Bahamas. That's limited to around 30 students, I think, and there's a small surcharge because they need to take a chartered plane, and they do field work out in the Bahamas there. But we have more closer to home as well. So we have a fieldwork module that runs in the summer, which is based in the UK. But yes, sadly, no chemistry fieldwork.
Yeah, thank you. And our last question is, what kind of research opportunities are available for undergraduate students in the biosciences program at Exeter?
So as well as the final year project, we recognize that more and more students are coming to university, not just because of an initial curiosity about a subject, but also because they want a job at the end of it. We understand that degrees are expensive pursuits. So we've worked very, very hard to create our internship program. So many of our academics offer internships in the summer for mostly for second year students entering their third year. Not always the case. Sometimes it's first years going into second year. And these are externally funded. So it's not like we're just asking a student to mop the floors and clean the windows or anything like that. It's projects for which they have applied for grants specifically for undergraduates to attend. So you've got those, you've got your final year project, and you've also got this industrial experience year, which sadly I didn't have much time to talk about, but there is information on the website. And as Rob said, I'd happily answer any questions via email. Just before you finish off, Zeynep, I have to say, these questions have been absolutely excellent. I've given a few of these talks in various guises now, in various countries, and I'm very happy to say that you guys ranked #1. So thank you for pushing us.
Yes, thank you very much. It was a very informative session for the attendees, and you covered all of the questions. Thank you for your answers as well. And also I would like to thank the participants in Turkish too. Katıldığınız için teşekkür ederiz arkadaşlar. Umarım sizin için de faydalı bir webinar olmuştur. University of Exeter ile ilgili diğer sorularınız için chat kısmında paylaşılmış olan mail adresinden International Office yetkililerine her zaman ulaşabilirsiniz. Bir sonraki webinarlarımızda görüşmek üzere. Thank you very much again, Sam. It was a pleasure to have you in IEFTalks.
Thank you very much. Have a good evening.
Thank you. Take care. Bye-bye.
Goodbye.